Welcome back to Vaccine Design course. In this lecture, we'll discuss how to predict the other types of epitopes such as discontinuous epitopes, then how to predict the immunogenic regions in a protein structure by going to another server, and then how to predict the glycoprotein antigen epitopes. So the first server that we are going to deal with is the Discotope server for predicting the discontinuous epitopes from the 3D structure of proteins in the PDB format. Okay, so for that we need to enter the PDB and chain in the options when you go to Discotope server. Okay, we'll be going to that, but we need to have the PDB and chain for it, right? So for that you need to do the BLAST P, or you can say protein blast, which is used for both identifying a query amino acid sequence and for finding similar sequences in protein database. So first, before going to Discotope server, let's go to NCBI protein blast page. Okay, so go to this particular link. You can see in the slide this particular link, that is blast.ncbi.nlm, and then you need to enter your FASTA sequence. So here I have searched for NCBI protein blast and click on to the first result. So this is your protein blast page. Here you need to enter your FASTA sequence. Okay, so which we already have. It's chain as spike glycoprotein. Copy it from the notepad and paste it here. Now since we want to see if it is there in the database, you need to change the database from non-redundant protein sequences to protein data bank proteins, which is PDB. Okay, then make others default and click submit. Okay, what I mean by default is you have the job title which is already there. Next, what we need the algorithm to be selected is BLAST P, which is already selected. Protein blast. Why we are doing is to identify the query amino acid and to find the similar sequences in protein database. We need to see if this particular sequence is there in the protein database to enter the PDB and chain ID in the Discotope server. Okay, so click on the blast button. After clicking that, you should wait for your results. Here in the results page, you can see the job title and the other information about it, the query length of it. And most importantly, you can see description, graphic summary, alignments, and taxonomy. So let's start by one. In description, here maximum score and the total score which is written here, this will tell you about the alignment. Now next what you can see is the query cover, which is the percentage of the area or length covered for the alignment. 100%, which is pretty good. E-value means it's the error value, and ideally E-value should be 0. So if you want to choose the one which has maximum query cover, then percentage identity, and then error value as 0. Okay, so that's the protein that you need to choose after getting the result of protein blast. Okay, so our hero here is percentage identity, because so let's arrange it based on the percentage identity. So for that, click on the percentage identity. Okay, so if you click on that, you can see that as the blocked one, and it will arrange according to the percentage identity. So it's 100%, pretty fair and good. So prioritize on query cover and percentage identity. Thankfully both are 100%. Okay, so let's go with the first one because that's the best out of all. So for that, what you need to do is you need to click on the accession number. Remember, it's 4xAKA. If you remember, when we did a particular thing, what we saw was the best template which was seen was 4xAK. Okay, even here when we did the blast P, it showed 4xAK as the best template. Okay, so let's click on the accession number first. Okay, so this particular number is actually the PDB ID. Now click onto it, then you will be directed to a PDB where you need to go for its FASTA. Okay, now it's chain A spike glycoprotein. Click on FASTA. So when you click on FASTA, you need to copy the PDB ID and chain ID. Okay. So since I've said that in the Discotope server, you need the PDB ID as well as the chain ID. So now the PDB ID here would be 4xAK and the chain ID is again A. So you can either do this procedure or directly look at the accession number and write it down somewhere. Okay, I just wanted to show you how normally it goes. Okay, it's straightforward. You can also just use the accession number and then you'll be able to know about it. Okay, next let's go to the Discotope server by going to this particular link. Okay, Discotope. For that, I'm searching as Discotope server in Google and click on the second result, which goes to toolsiedb.org. Yeah, so here it says to enter the 4-letter PDB ID, or you can upload the PDB file. So since we have the PDB ID, we are entering 4xAK, and the chain ID, we all know that it's chain A. Now it's just the version of it. Then you can click the submit button. Now here, as you can see, the threshold that they have marked was -7.7. You can actually change this. You can have a table view, 3D view, and you can actually save the prediction as well. Okay, so let's first go to the table view option. Okay, then so this is the table view. Now you can always click the help button to know how it is predicted. See, everything is mentioned here, how the table view is getting interpreted, how the 3D view is getting interpreted. Okay, so this is the table view where the interpretations, everything is given: chain ID, residue ID, the contact number, propensity score, and the Discotope score. So the two heroes that we need to know about is the propensity score and the Discotope score. Please have a read about it. Okay, now let's come back to the table view. Okay, so now to talk about the propensity score, it tells you the efficiency of the epitope. Okay, the first one means it's more efficient. That's how it goes. Now Discotope score, it is calculated by combining the contact numbers with the propensity score. Okay, so the Discotope score above the threshold value, which was, how much was the threshold? It was -7.7, I guess. Yeah, so the score that is above the threshold value indicates positive predictions, and that below it indicates negative predictions. So positive means they are efficient of being an epitope, and negative means it's vice versa. Let me just look. Let me just click here. Yeah, so just click here, then only you'll be able to see the efficient epitopes, okay, which are marked in green. Okay, that's how you need to click. Okay, it's not arranged automatically. You have to click onto it to see which are the efficient epitopes. Now if you go to the chart view, so the efficient one, the first one would be, yeah, based on the, so let's just, yeah, so the propensity score is calculated. It tells you the probability of being part of an epitope. Discotope score above it indicates positive prediction. Okay, so that's how you need to interpret. Now let's go to the 3D view, which gives you a better interpretation. So this is the 3D view. This is the model structure. Now let's just click the CPK. If you click the CPK, then what it shows is the residue will be shown in the model structure. The next CPK, this is where the next residue comes, the third residue, the fourth residue, and so on. Okay, now when you go for the chart view, that is actually a better interpretation. Okay, so the chart view where you can see the threshold -7.7 and the positive prediction. So these are all the positive predictions, and the table view indicates the different residues, which says specific residues. Okay, so these are the epitopes. Okay, that's what all these pink ones are, negative ones. Now, as I've mentioned, there is something called a save prediction where you can save it by clicking on it. Okay, now the next server that you can explore is the Epitopia. Okay, so this was just the results page, how it is predicted and stuff. Let's go to the next server which is named epitopia.